Researchers and physicians have been studying dysbiosis or alteration in gut flora and many disease processes, including inflammatory bowel disease, irritable bowel syndrome, rheumatoid arthritis, depression, diabetes, metabolic syndrome, multiple sclerosis, Parkinson's disease, chronic fatigue syndrome, and heart disease. Much research in recent years has focused on fecal microbiome transplant, or FMT, as a potential therapy for disease prevention and to treat numerous medical conditions beyond C. diff. For those unfamiliar with FMT, stool can be transplanted by colonoscopy, enema, frozen pills, and even nasoenteric tube. Number 1, government regulation. There are several conditions that can contribute to dysbiosis, including antibiotics and diet. Our ultimate goal is to use FMT to improve the fecal microbiome and to cure a variety of disease processes. While we lack data about the long-term side effects of FMT, animal models have shown that FMT can unintentionally transfer disease phenotype, including metabolic disorders and obesity. Gut bacteria have also been accused of increasing the rate of both colon cancer and heart disease in patients. So it's very important that FMT be regulated properly. The U.S. Food and Drug Administration works to make sure that patients have safe access to treatments that are effective through research and evidence-based medicine. As explained in a recent American Journal of Gastroenterology article, which I helped contribute to as a member of ACG's FDA-related matters committee, quote, in the United States, drugs and bioactive agents, including microbiome-based treatments or MBTs, used with therapeutic intent for specific disease states, falls under the jurisdiction of the U.S. Food and Drug Administration, end quote. The Center for Biologics Evaluation and Research, or CBER, is the part of the FDA that regulates biologic products, which itself includes blood as well as blood products, vaccines, gene therapies, allergenics, plus, quote, human cells and tissue products, end quote. The FDA previously decided that the fecal microbiota is both a drug and a biological product and should be regulated as a drug. Now, interestingly, the Federal Food, Drug, and Cosmetic Act, or FDCA, defines drugs as, quote, articles intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease, and articles other than foods that are intended to affect the structure and function of the body of man or other animals. And now, in addition, the Public Health Service, or PHS Act, says that a biological product is, quote, a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, allergenic product, protein, or analogous product applicable to the prevention, treatment, or cure of disease or condition in human beings. As such, biologic products in the United States can be regulated as drugs under the Federal Food, Drug, and Cosmetic Act, or as biological products under the Public Health Service Act, or under both laws. Clinical trials of microbiome-based treatments require an investigational new drug, or IND, application, which is basically a request to the FDA for permission to give a potentially therapeutic microbiome donation to humans. Advantages of the FDA. One of the advantages of having the FDA regulate FMT is that the industry can more easily navigate their medical legal responsibilities through a uniform, organized process when bringing fecal microbiome product to market. The FDA can also help physicians more successfully treat patients by providing guidance. Number 3, disadvantages of the FDA regulating. It's difficult for the FDA to regulate potential do-it-yourself treatments when hospitals and scientists run their own stool banks or utilize donor stool from patients. Unlike medications that are produced with strict standards in a factory using controlled amounts of standardized ingredients, each human stool has a unique microbiome makeup that can also evolve over time as donors age. So it's harder to regulate donor stool for FMT. Microbiome diversity. It's difficult to regulate FMT due to the complexity, the number of bacteria in the microbiome, and because the microbiome of each donor is different. When hospitals or stool banks create FMT product, the unique microbiome components of each FMT product would need to be identified, purified, and tested. Number 5, success in standardized stool banks. Fecal microbiota transplant, or FMT, is a very effective treatment for recurrent C. diff colitis when antibiotics aren't resolving the infection. FMT has been supported by multiple society guidelines. Over time, the introduction of standardized stool banks has made access to fully screened donor stool extremely accessible for physicians nationally. What's interesting is that the FDA still considers FMT an investigational new drug, and physicians and scientists need to submit an IND application. Since FMT is still considered investigational by many regulatory agencies, insurance payment for C. diff treatment can be hindered sometimes. Recently, pharmaceutical companies have received FDA approval for 2 new microbiome-based therapies. Rheumatoid, made by Ferring Pharmaceuticals, was designed to prevent C. diff recurrences in patients 18 years of age and older. Unfortunately, many C. diff patients experience a recurrence after the first antibiotic for C. diff treatment, possibly due to dysbiosis. Rheumatoid helps restore gut flora. It can't be used until after the first episode of C. diff. Rheumatoid is delivered rectally from donor stool. In terms of regulation, Rheumatoid was given priority review, orphan drug, and breakthrough therapy FDA designations. In addition, VOWST was FDA approved in April 2023 and is delivered orally. VOWST is important because it's the first oral microbiome-based therapy used to prevent C. diff. Patients take 4 pills per day for a total of 3 days. Gastroenterologists have welcomed both new drugs as new tools in our armamentarium against C. diff, which can cause diarrhea, leading to death in certain severe cases. Side effects and possible complications of FMT. In general, FMT is very safe, yet there are potential side effects which can range from mild stomach upset to a few cases of severe infection. Being immunocompromised was a risk factor. Reported GI side effects have included crampy abdominal pain, bloating, flatus, belching, burping, diarrhea or constipation, nausea, sometimes vomiting, and increased borborygmus or stomach rumbling. Unfortunately, there has been a severe case of E. coli infection that resulted in death, which prompted the FDA to mandate that all stool be screened for E. coli infection. The patient who passed away was receiving FMT for an experimental treatment of allogeneic hematopoietic cell transplantation using a hospital-based stool bank that was not screened for multi-drug resistant organisms. This case sounded alarm bells in the medical industry. Yet, FMT in general is extremely safe, and because the potential success for disease modification is so great, many researchers and gastroenterologists hope that one day soon we may use FMT for many medical diagnoses. Since the E. coli patient with the allogeneic hematopoietic cell transplantation in 2019, the FDA has provided a required screening process to improve safety for patients. The most important lesson from these incidents was to standardize stool donor collection even further. Moving forward, FMT should theoretically be safer and safer with increased screening protocols. Administering FMT by upper gastrointestinal delivery can place patients at risk for aspiration. Unfortunately, in the past, there was a patient who died of fecal aspiration, so it's important to provide an antiemetic medication when delivering it via upper scope. Also, these patients should not be sedated. Delivery via colonoscopy places patients at slightly increased risk of perforation. It's important to have a thorough benefits and risk conversation with the patient in advance and assign consent detailing all known potential side effects of FMT. Number 7, protecting the public. I recommend a stricter standardized testing of donor stool and blood prior to transplantation in order to prevent any infection risk. One author has recommended that HIV testing be performed 14 days after donation to help further eliminate any risk. Perhaps a risk assessment should be performed to prevent FMT donation from individuals who are engaging in risky behaviors. We should consider how FMT is stored and its expiration life. For instance, there are certain organisms such as Faecalibacterium prausnitzii frequently found in inflammatory bowel disease patients which can be damaged by oxygen. So careful attention needs to be paid to how to prevent oxygen exposure to FMT donations. We may need to regulate storage conditions, including storage temperature and how stool is stored. Lots of future safeguard research should focus on potential side effects of FMT for recipients. Also, we need to determine whether donors who eventually develop significant medical problems such as cancer later in life are impacting recipient patients through donor stool. FDA orphan drug designation. Another area of concern as FMT therapeutics takes off is the FDA orphan drug designation could prevent competing products from coming to market unless the drug production process is novel and completely different. Greater attention should be placed on how FMT is prepared. Number 9, role for IRB and bioethicists. As research into FMT continues, I highly recommend that institutional review boards work together with the FDA. Bioethicists should remain involved due to the potential downstream effects of microbiome manipulation during research. Researchers should work together with the IRBs, the FDA, and bioethicists to determine which are acceptable risks for patients. Within FMT research, systems should be placed to help track long-term health outcomes for all patients and studies. The future of fecal microbiome transplant being used to alter the course of disease is extremely promising. Some questions that an excellent recent article in Nature magazine written by Kirsty Agard and Elizabeth Homan proposed, quote, what duration of follow-up is necessary? How might microbial changes be altered by future drugs, diseases, or age? At the completion of a study in which participating subjects are given live microbial therapeutics, should these subjects receive antibiotics for eradication of the study microbe, regardless of study findings? And if so, what collateral damage might such antimicrobial therapy subsequently unleash? End quote. These are important questions that bioethicists and the FDA will need to help regulate. As we research the ability to alter the fecal microbiome, it would be important to make adjustments, fine-tuning treatment to account for diet and host genotype. Number 10, regulation and tracking donors. Perhaps human stool and FMT should be recategorized within its own unique category of the FDA and internationally for regulatory purposes. Stool banks and national registries should work together for long-term tracking of donors and recipients over time. As FMT products are brought to the market in the future, companies should use manufacturing guidelines to help make sure that each step of the manufacturing process is uniform for patient safety.